This method can be used to prepare biomolecule activated surfaces, which have applications in areas such as drug delivery, biological target detection, and bioseparation. The main advantage of this technique is that the poly-PFPA brushes are highly reactive with amines, and the immobilization of antibodies is achieved by incubation in buffer solution for a few hours.
The implications of this technique extend toward the protein purification through immunoprecipitation, as the immobilized antibody can selectively bind with target antigens.
Though this method is demonstrated for antibody immobilization and protein purification, it can also be applied to other systems requiring biomolecular immobilization.
To treat silicon dioxide beads with APTES, first obtain silicon dioxide particles in the form of 5% weight volume aqueous suspension. Combine 0.8 milliliters of silicon dioxide suspension with 40 milligrams of APTES and 8 milliliters of methanol in a 20 milliliter scintillation vial equipped with a stir bar. Allow the reaction to proceed at room temperature for 5 hours with vigorous stirring. After 5 hours, transfer the solution to a conical tube. To isolate the APTES functionalized silicon dioxide beads, centrifuge the solution at 10,000 G for 5 minutes. Then remove the supernatant. Now wash the beads by redispersing them in 3 milliliters of fresh methanol. Shake the tube by hand for mixing, but if necessary, improve the dispersion by sonication in a water bath for a few seconds. Centrifuge as before.